Hello everyone, welcome to our webinar entitled Explore Vascular Function in Vivo and at Cellular Resolution with Cellvizio Lab. My name is Elodie Lee and I am Product Marketing Manager in the Preclinical Business Unit. So today we will discuss about applications of Cellvizio Lab in vascular imaging and in particular I will show you some examples where the technology has been used to study the anti-angiogenic effect of a drug, to look at extravasation, and to characterize microcirculation. During the presentation, do not hesitate to type your questions or comments in the Q&A box, which is located at the bottom of your screen. I will take your questions during the live Q&A session after the presentation. So now let's listen to the presentation.
During this webinar, I will discuss about applications of Cellvizio Lab for in vivo vascular imaging. In the first part, I will give you an introduction of the technology and give you some examples of applications where Cellvizio Lab was used to characterize vasculature in vivo. Then in the second part, I have selected three scientific articles that illustrate the use of the technology to characterize vasculature for different applications. The first application would be the study of the anti-angiogenic potential of a molecule. Then we will discuss how Cellvizio Lab can be used to understand drug activity through vascular imaging. And the last example is more a translational application of Cellvizio, where it was used to characterize the microvasculature in a sepsis model. So, about the technology, Cellvizio is a fluorescence-based confocal laser endomicroscope. That technology can be used to image any type of tissue with cellular resolution on the living animal. So what you see on the image is a bundle of optical fibers that we use. We put that bundle in contact with the tissue of interest to get that image. So the technology is being used in preclinical research. This is our instrument, Cellvizio dual band, for certain different types of applications in different fields of research, like cancer research, infectious disease, drug discovery, stem cells, etc. But the technology has also been cleared for the clinics for certain indications, in particular to analyze the digestive tract, pulmonary tract, and urinary tract. So regarding the preclinical instrument, the instrument has two lasers, 488 and 660 nanometers, to excite molecules and to collect simultaneously the fluorescence emitted by these molecules. The confocal microprobe, so the bundle of optical fibers, is connected to this laser scanning unit, and you have a different range of confocal microprobes depending on their tissue of interest. These confocal microprobes are flexible, so you can combine them with an endoscope or use them in combination with needles or catheters to have a minimally invasive access to the tissue of interest. So, depending on the application, depending on the biological phenomenon that you want to image, you can choose the best fit for your application. You have three types of confocal microprobes. The S series for surface imaging, M series, those microprobes will image with a higher spatial resolution, down to 1.4 microns, and the Z series to image much deeper into the tissue, from 100 to 200 microns from the point of contact. So what are the applications of Cellvizio for vascular imaging? The first example is to use Cellvizio to look at angiogenesis. For example, to locate biomarkers of angiogenesis or to image tumor neovessels. So on the video on the right, what you see is a video recorded on a subcutaneous tumor on a mouse. Tumor vessels are highlighted in green with the use of fitsidextrin, and angiostamp was used to look at integrins that are specific in angiogenesis. The second example is on the left, the tumor neovessels, highlighted again with the fitsidextrin injected through IV. And you see those tumor vessels that are very disorganized and very small tortuous. And the video on the right is an image collected on the same subcutaneous tumor after a nanobody was injected. So this nanobody was targeting HR2. And so you see at the same time the localization of the nanobody and the tumor vasculature. Another application of Cellvizio for vascular imaging is looking at extravasation. Extravasation can give information about to confirm the drug delivery or to confirm the efficacy of treatment. So the example on the right shows extravasation. So you see here a tumor vasculature highlighted in green with the fitsidextrin at 1 hour, 4 hours, and 24 hours after the injection of the tracer. And these are labeled nanoparticles that fluoresce with the red laser, again 1 hour, 4 hours, and 24 hours after the injection. And the third column is the merge of those two images. So what you see here is the biodistribution of those nanoparticles in the tumor, and you see that those nanoparticles at 4 hours they remain in the blood vessels, but after 24 hours they extravasate. Another way to look at vasculature using Cellvizio is to measure some morphological alterations that are due to disease development or due to treatment that you are applying. And measuring this morphological alteration helps you better understand the development of the disease or the action of a drug. And these type of techniques can be used to better translate the methodology to the clinics. And in particular, we will see some applications of our module. It's a plugin that is called Vessel Detection that is used to get some quantitative information about the vasculature. So the first example that I wanted to show you is the study of the anti-angiogenic potential of a molecule. So that was an article published by a team from Hong Kong. So the study focuses on endometriosis. Endometriosis is a chronic disorder that is characterized by the implantation of endometrial glands and stroma, but outside of the uterine cavity. And this disease affects 1 in 10 women on average in the world. And what is the particularity of endometriosis is that it is an angiogenesis dependent disorder. Angiogenesis plays a very important role in the growth and in the survival of those endometric lesions. So the idea is to see whether anti-angiogenesis therapy could offer a new opportunity for the treatment of endometriosis. And in this study, the article focuses on EGCG, which is a polyphenol that is found in green tea, and it has been shown that EGCG has antiangiogenic properties. So the study focuses on characterizing EGCG, but also a synthetic derivative of EGCG, which is called pro-EGCG. The fact that this molecule is being synthetic is that it is more stable and more easy to produce. So here we want to know whether pro-EGCG could be a potential and novel anti-angiogenesis agent for endometriosis. So first, they used whole body bioluminescence imaging to monitor the growth of the lesion during 4 weeks on the same animal after applying different sorts of treatment. So what you see here is the baseline at the beginning of the treatment. And these are images taken after 3 weeks or after 4 weeks of treatment. The first row is a control where only saline was injected. Here is vitamin E that doesn't have any antiangiogenic properties. Here are the 2 molecules of interest, EGCG and pro-EGCG. So what can we see after 4 weeks is that after EGCG treatment, the bioluminescence from the lesion is lower than the bioluminescence from the saline group. So you look at here, starting week number 2. And this is even more characterized after 4 weeks. As for pro-EGCG, you see also that the lesion size is smaller than saline, but also smaller than EGCG. And those results, those bioluminescence results, were confirmed by measuring the size of the lesion and the weight of the lesion after terminal surgery. So what you see here are the lesions here, okay, after the different types of treatment, and the graph here show the lesion size and weight after terminal surgery. So you see here that there is a huge effect of pro-EGCG on the size of the lesion and the weight of the lesion. So then bioluminescence helped confirm that pro-EGCG has a very positive effect on preventing the development of those endometric lesions. But how about the effect on the vascular structure? So to do so, the users use Cellvizio imaging to have a closer look at the vasculature to be able to characterize the vasculature, and they combine Cellvizio imaging with micro-CT imaging ex vivo to have a 3D view of the microvasculature. So what you see here is the vasculature as imaged using Cellvizio after fitsidextrin was injected. And what can be revealed is that you see less vessels when EGCG or pro-EGCG are administered compared to saline or vitamin E, and those effects are amplified when using pro-EGCG. So you can see here on the images, but the users also compared, for example, the functional capillary density or the total vessel length across those four types of treatment. The users also looked at cell death apoptosis using YoPro1 that was directly injected in the lesion area, and they used a higher resolution confocal microprobe, a Minio 100. And what they did is that they counted manually the number of cells that could be labeled using this dye YoPro, and they found that you have an increase of apoptosis with EGCG and pro-EGCG treatment. And again, those effects were amplified using pro-EGCG. So to conclude with this article, it is a nice example of showing the complementarity between different imaging modalities. First, you can use whole body optical imaging to study the development and the growth of a lesion, and use Cellvizio imaging to analyze the antiangiogenic effect of a potential drug candidate. And in this particular study, they got a confirmation that pro-EGCG could be a potential antiangiogenic molecule for the treatment of endometriosis. So the second example I wanted to show you is the study of different doxorubicin polymers and how these doxorubicin polymers, can inhibit angiogenesis. So we are going to talk about RGD peptides. RGD stands for arginine, glycine, and aspartic acid. And it has been shown that these peptides have a very high affinity with alpha-V integrins. And these integrins are overexpressed in angiogenic vessels and in tumor cells. So the idea is to use those RGD peptides as vectors to deliver doxorubicin as polymers directly to the tumor. So we are going to talk about RGD PPCD, which are doxorubicin coupled to RGD peptides. And some previous studies have shown that those doxorubicin peptides, they can accumulate around tumor blood vessels, but they cannot penetrate the tumor. So if you look at the drawing at the bottom, you see RGD PPCD, so the doxorubicin polymers in green, and you see that they can remain in their they accumulate around the blood vessels, but they cannot penetrate the tumor. So the users have developed another a derivative of those RGD peptides that they have called internalizing RGD peptide, or IRGD. And the hypothesis is that IRGD could increase the penetration into the tumor thanks to the activation of neutrophils. So here again, the same drawing, you see IRGD PPCD, so the internalizing peptide in red, and you see that it can accumulate around blood vessels, but also it can penetrate into the tumor. So the aim of the study is to better understand the mechanisms that are involved in the delivery of doxorubicin using RGD and IRGD peptides, and to study the effect of those doxorubicin polymers on angiogenesis. So to do so, they used a mouse model, a subcutaneous tumor, and the different types of treatment of doxorubicin were administered through IV once per week during 4 weeks. And after treatment, at day 25, the Cellvizio analysis was performed. First, to look at vascular permeability and extravasation, and also to look at vascular morphology. So to do so, they used 2 different types of confocal microprobes. Regarding the analysis, they also used a lot vessel detection, which is a plugin that we have developed that is based on the segmentation of the image that you collect using the Cellvizio instrument. And based on the segmentation, you can collect some quantitative results about the vasculature, such as the total length of the vessel, the mean diameter, or the functional capillary density. So first, looking at extravasation, they use the Cellvizio to monitor dynamically extravasation of different doxorubicin polymers. So to do so, they looked at the vasculature by injecting fitsidextrin in the vasculature. And they monitored the vasculature on the same animal longitudinally at 4 different time points: 1 minute, 5 minutes, 10 minutes, and 20 minutes. Different treatments were administered, just the control, saline, doxorubicin itself, the doxorubicin polymer, the doxorubicin polymer co-administered with the RGD peptide, or the doxorubicin coupled to the RGD peptide, and the same for IRGD peptide. And what can be seen is that after 20 minutes, the boundaries of the vasculature cannot be clearly distinguished when IRGD peptide is used. They also looked at the permeability by looking at the signal inside and outside of the vessels at different time points, and they observed that they had a higher permeability when IRGD was coupled to the doxorubicin polymers. So that was the effect on extravasation. How about the effect on angiogenesis, on the morphology of the tumor vasculature? So to do so, they compared again the vascular morphology of different mouse models that were administered the different types of treatments. To do so, they injected fitsidextrin and looked at vascular density and vascular diameter. What they observed is that when the doxorubicin polymers are coupled with RGD peptide, they had a decreased vascular density and a decreased vascular diameter compared, so look at here, compared to saline. So that is the vascular density, and here this is the vascular diameter. And again, they noticed that when using IRGD, internalizing RGD peptide, those angiogenic effects were enhanced. So to conclude with this study, the authors have shown using an in vitro study, in vitro experiment, they observed that there was a higher uptake of doxorubicin polymers when those polymers were coupled to IRGD peptide. But with the Cellvizio imaging, they observed that the reasons why you had this higher uptake. First, it was because the vascular permeability was enhanced using this IRGD peptide, and also because the doxorubicin polymers, they inhibit tumor vascular growth. So what I want you to remember, what are the benefits of Cellvizio imaging for this particular case, is that it makes it possible to understand the activity of a drug in vivo, and you can study the response to drug treatment directly in live tissue. So the last example that I wanted to show you is more a translational study, so an application of Cellvizio in preclinical research, but on larger animals. So here we are going to look at the microvasculature in a sepsis model. Microcirculation alteration are common in sepsis, and they play an important role in the pathophysiology of sepsis-associated organ dysfunction. So we see that improving the microcirculation could be an emerging therapeutic target in the treatment of sepsis. But that would require the bedside monitoring of microcirculation. So the objective of this study was to evaluate the feasibility of Cellvizio to image in vivo microcirculatory alteration in various GI mucosal beds, but also to quantify the effects of volume therapy on the early stages of the septic shock in a pig model. So in this study, they used the clinical instrument. You will see why. So the clinical instrument was used. So first on the preclinical models, so on pigs, and the instrument was used in combination with an endoscope, and different mucosal regions were analyzed just before the initiation of the septic shock, during sepsis, and after volume therapy using gelatin. In a second study, they initiated a test, a study directly on patients. So that is the reason why they used the clinical instrument. So they made the analysis on 10 patients with severe sepsis and 8 healthy controls. And for this application, they imaged the duodenal mucosa. So these are images that were obtained in different areas of their GI tract. So the gastric antrum, the duodenum, and the rectum. As I mentioned earlier, so they used the plugin vessel detection to get to have first an automatic detection of the vessels, but also to get automatically some measurements, some quantification of the vasculature, just such as the length of the vessels, the mean diameter, or the functional capillary density. So in the setup of the study, the images, the Cellvizio images, were obtained at baseline, during the septic shock, and after volume therapy. And they compared those different characteristics of the microvasculature. So what they observed is that the functional capillary density reduced in all the different segments of the GI tract during the septic shock. But after volume therapy, this FCD was improved. Regarding the characterization of the microcirculation in humans, They observed that there was no significant difference in the total vessel length between healthy and septic patients, but they observed a decreased vessel diameter and decreased functional capillary density in septic patients. So here, in conclusion, I want you to keep in mind that the direct assessment of the microcirculation is made possible using Cellvizio technology, and they could observe some heterogeneity in microvascular perfusion in different GI mucosa. The volume therapy using gelatin improved the upper and lower GI mucosa in the early stages of the septic shock in pigs. So just to conclude about this presentation, I want you to remember the benefits of Cellvizio to look at vasculature in vivo. So first, you can use Cellvizio to study angiogenesis, to localize molecular markers of angiogenesis, or assess the impact of therapy on angiogenesis. You can follow in vivo extravasation to evaluate different strategies of drug delivery, or even to confirm the efficacy of the treatment. Or another application is to characterize microcirculation in vivo. You can measure morphological alteration that can be due to the disease or the treatment, and you can translate that methodology to the clinics quite easily. So thank you very much for listening to the webinar, and I will now be able to answer your questions.
So now I will open the Q&A session. So do not hesitate to type your questions in the chat box, which is located at the bottom of your screen. I will take questions as they come. So I have a first question regarding fluorophores. So what types of fluorophores can we use to look at vasculature? So I gave you some examples during the presentation. So one that is very common is FITC that is coupled to dextran. So dextran is a biopolymer, and you can choose the size of the dextran molecule for your experiment, depending on what phenomenon you want to image. If, for example, if you want to just highlight vasculature, you can choose a high molecular weight dextran so that it will remain within the vasculature. If you want to look at phenomena like extravasation, then you can choose a smaller dextran size so that it can extravasate. So FITC dextran, you can inject it using IV, and it will fluoresce with the 488 nanometer laser. Another possibility is to use Evans blue. Evans blue will work with the 660 nanometer laser. So you will see that on the red channel. And Evans blue binds to albumin. There are other commercial dyes that you could use to look at vasculature, such as AngioSense or AngioSpark, for example. I have another question regarding blood flow. Okay, can we measure blood flow using Cellvizio? So we are not able to measure the blood flow in real time. However, on the videos, I showed some videos at the beginning, it is possible to see blood flowing. And just keep in mind that our standard acquisition rate is between 9 to 40 hertz. So it will you will need to check whether this acquisition rate is compatible with the events that you want to measure. And so the webinar is being recorded at the moment. So you will receive a link to watch the recording at a later stage. So let me check with other questions. So I have a question regarding the use of antibodies. So we have some examples where antibodies have been used so that you can detect some specific markers. And of course, these antibodies have to be previously labeled with the fluorophore. And these fluorophore need to be excited with our 488 laser or 660 laser. So for example, there are some commercial dyes that are available to label antibodies like Alexa fluor dyes or let's say IR dyes, things like that. So another question. I don't know if you have more question. I have another question regarding endovascular imaging. So endovascular imaging means to be able to insert our confocal probe inside the vessel. So of course, the larger the animal, the easier the procedure. For example, if you want to work on the mouse, endovascular access would be difficult because the vessels are very small. Keep in mind that our smallest microprobe has a tip diameter of 300 microns. So you need to check whether it is compatible with the vessel you want to penetrate. So for rats, maybe it's a little bit difficult, but we have done some tests with rabbits and it worked fine. So now that the presentation is finished, I will open the Q&A session. So I have already some questions, but do not hesitate to type your questions in the chat box, which is located at the bottom of your screen. So I have one question regarding wavelength. So we have two wavelengths with a dual band instrument, 488 and 660 nanometers. These are available in the standard configuration. For other wavelength, you should contact us, but that it requires some development. So in the standard configuration, it is only 488 and 660 nanometers. Then I have another question still regarding the different channels. So whether it is easy to switch from one laser to another laser. So actually, our system will send the two laser wavelengths simultaneously. So you have a set of mirrors that are used to scan the tissue using the confocal microprobe, and both lasers are scanned simultaneously. So at the same time, you have the 488 and the 660 lasers. And in terms of images, you get real-time images in the software of both the green channel, so the 488 line, 660 line, and also the merge of the two lines. And there is no white light image in the instrument. We only collect the fluorescence signal. So I have another question regarding the type of fluorophores that we can use to look at vasculature. So I have given some examples during the presentation. So you can use, for example, dextran. So dextran is a biopolymer that is which you can select the size, and you can link it to FITC, which will fluoresce with the 488 line. And depending on the phenomenon that you want to image, if it's extravasation, for example, you can choose the molecular weight of the dextran molecule. If you are looking only at vasculature, then you can select a high molecular weight dextran so that you can be sure that it will remain within the vasculature. So that is for the 488 line. For the 660 line, you could use Evans blue, for example, and Evans blue will bind to albumin. There are other commercial dyes like angiogenesis that you can also inject through IV. So I have another question regarding specific application looking at the blood-brain barrier and whether our technology can be used to look at the cerebral vessels. So yes, of course, you can work with our instrument to look at the blood-brain barrier. So again, you need to, if it's, let's say, at the surface of the brain, you need to give access to our confocal microprobe so that the confocal microprobe can touch the brain. It needs to touch the tissue, and then you could image vessels that are located in the cortex area, for example. And then if you want to look at vessels that are located inside the brain, this is still possible. We have a very recent publication from a lab in the US from Professor McNeek, who is looking at hippocampal vessels. In that case, you can use a very thin confocal microprobes that have a beveled tip, so that these confocal microprobes can be inserted deep into the brain. So I can send you the reference for this type of article. So if you have more questions, do not hesitate to type your question. I have another question still regarding blood flow. Are we able to measure blood flow? So with our instrument, we are not able to actually measure the blood flow, but on the video we can see blood flowing. Note that the acquisition rate of our instrument is between 9 to 40 Hertz. So then it will depend on what event you want to measure, whether it is compatible with our acquisition rate. So I have another question still regarding exploring deep brain vessels. How can we insert the probe in the hippocampus? So what you can do is, if you are working on the anesthetized animal, you can use a stereotaxic frame, open the skull of the animal, just a small opening, and then the probe can be inserted. So using the stereotaxic frame, using very precise coordinates to the specific area that you want to image. I don't know if you have other questions coming up. I will wait just 1 or 2 seconds for more questions. If there are no more questions, I will close this Q&A session. If you have more questions, don't hesitate to send them by email, and I will get back to you after the webinar. One last question, for example. So one question regarding our confocal microprobes. If there is a specific probe that we can use to adjust for the Z movement. So I guess it is whether we can change the confocal plane of our probe. So in our configuration, the confocal plane is fixed. So it means that you are not able to do some Z-stacks, for example. Okay, what we will image is a single plane. It's a confocal plane, and it is fixed depending on the microprobes. We have different sets of microprobes. Some will look only at the surface, so from the point of contact, from 0 to 15 microns, let's say, from the point of contact. These are surface probes. And to look at deeper areas, we have the Z probes. The Z probe would look at, let's say, roughly 150 microns from the point of contact. So you need to touch the Z probe with the tissue, and what you will image is underneath. So let's say more than 100 microns from the point of contact. Okay, and I have other questions. So whether or not the resolution of our instrumentation is enough to image bacteria. So yes, the resolution that we have is is a cellular scale resolution. Depending on the probe that you are using, you can have from 3.5 to 1.4 microns. So you will be able to detect some bacteria, individual bacteria or individual cells. What you will not be able to do is to look at compartments, okay? What is inside the cell. And another question, I think it's more related to the experimental protocol. It is true that blood vessels there are some dynamics and some movement. How can we compensate for that? So it is in the analysis, in the analysis, the way you will analyze the data. For example, if you want to look at vascular constriction, so you are able to measure the vessel diameter, but if you want to get a confirmation that what you see is a true decrease of diameter, then you will have to check to have some internal controls of vessels that will help you confirm that what you see is really a constriction and not. So now I would like to thank all the participants for joining us today and for making this very interactive. I hope that you enjoyed the content of the webinar. And if you have more questions, do not hesitate to send your questions by email, and we will get back to you very shortly. So the webinar was recorded, and you will receive next week a link to watch the recording of this webinar. Thank you again, and bye-bye.